we are in the metaplasia, we are changing the cell from one form to another. We are changing the cell from one phenotype to another. Why? Because there's a stressor, and the cells that are initially there might not be able to cope with that stressor. So we need to change it to another cell type that is better at handling that stressor. That's the concept of metaplasia. But what is driving that changing or that replacement is due to reprogramming of the stem cells. It's due to reprogramming of stem cells. All stem cells, they are kind of dedicated to giving rise to some particular differentiated cells. Let's say we have like uh stem cells that produces that forms the the epithelium or the epithelial cells of the esophagus. And what those stem cells produce is just squamous cell epithelial cells, squamous cells. Now, if there's a metaplasia, you are going to have reprogramming of those stem cells, and they will no longer differentiate towards the direction of the squamous cells. They will now go somewhere else. They can go to columnar, they can go to cuboidal. If that one is going to cope with the stress, they will prefer to differentiate along that axis. That is metaplasia. Reprogramming of the stem cells to differentiate into another cell type, to differentiate from one cell type into another one that can adapt to a new stress. The common example for that. Now, before I move, metaplasia typically occurs in two forms of cells: epithelial cells and mesenchymal cells. Epithelial cells and mesenchymal. We are more familiar with the epithelial cells. Now we'll talk about both. For the epithelial cells, the commonest form of change is the Barrett esophagus. That's the one we commonly talk about. So in Barrett esophagus, we are just having a reflux of gastric acid from the stomach into the esophagus, maybe due to an incompetent lower esophageal sphincter. So there's a problem with the lower esophageal sphincter, and it cannot prevent gastric acid from refluxing from the stomach into the esophagus. So if it doesn't prevent that, that means that acid will be entering the esophagus. And the only way the esophagus can cope with that acid is to behave like the cells that is producing the acid, to behave like cells that are found around where that acid is coming from. The acid is coming from the stomach, and the stomach has epithelial cell that is similar to the intestine. That's the um, simple columnar epithelium. When there is reflux of the acid back into the esophagus is lined by stratified squamous epithelium. That stratified squamous epithelium cannot handle that reflux. So what will it do? Is the stem cells there are going to change their differentiation into the simple columnar epithelial cell. So that will make them undo that acid reflux. So when you're now looking at the esophageal wall, on biopsy, and we are now seeing areas of uh, stratified squamous upwards, and below you are now seeing simple columnar epithelium, that's likely to be a metaplastic change. So they are trying to cope with the new irritant, which is a gastric acid that is, refluxing to the lower part of the esophagus. That's what we call Barrett esophagus. Barrett esophagus. Another example is someone that smokes. So because of the fact that smoke, the content of the smoke, this carbon monoxide, the nicotine, the tar, everything irritates the lining of the airway. And what lines the respiratory tract, particularly the trachea and the bronchi, includes the pseudostratified ciliated columnar epithelium. That is what you find there, the pseudostratified ciliated columnar epithelium. But that one cannot handle the irritation of smoke. It cannot. What can handle the irritation of smoke is the stratified squamous epithelium, like the one you see in the esophagus. So that means that the epithelial cell lining the airway have to differentiate into that part. So we are going to have change from the stratified squamous epithelium to stratified the change from the stratified columnar epithelium to stratified squamous epithelium. That kind of change is a metaplastic change. It is important to remember that metaplasia and hyperplasia. Both of them can progress to dysplasia, and from dysplasia they go to malignancy. So that is why someone that is having Barrett esophagus due to reflux esophagitis, you must always screen them every time in order to ensure that they have not progressed to esophageal cancer. So you must always make sure that they have not, um, uh, progressed from the metaplastic change, for example, the Barrett esophagus now, which is simple columnar, to a dysplastic change, then to carcinoma. You want to ensure that it has not changed to carcinoma because there's a high chance it's going to get to esophageal adenocarcinoma, because it's going to take the epithelium of the glands. Stomach, they contain so many glands, so the epithelial cells there are in form of, when you see the word adeno, that means glandular epithelium. So it's going to go to adenocarcinoma. Yeah. So someone that is having tobacco smoke now, causing a metaplastic change of the respiratory epithelium from a pseudostratified columnar epithelium to stratified squamous. That kind of person can develop a stratified squamous carcinoma of the lung because of that. That is why smoking causes stratified squamous carcinoma, because they are differentiating along that stratified squamous epithelial pattern. Another form of metaplasia that happens outside the epithelial cells is metaplasia of connective tissue or metaplasia of mesenchymal tissue. We have a lot of connective tissue. Blood is a connective tissue, bone is one, muscle is another one. There are a lot of them. A form of metaplasia that can affect connective tissue is what we call myositis ossificans. So let's say somebody suffers a fracture and there is injury to the muscle. There's bleeding into that muscle. Instead of the stem cells of the muscles to regenerate into some new muscle cells, some of them will, be reprogrammed and they will differentiate along the axis of bone cells. So bones will be deposited on, those damaged muscles. I will call that myositis ossificans. So ossification, we are forming bone, myositis on the muscle. So once you see formation of bone within a muscle after trauma that is presenting like an hard mass, that is a metaplastic change, but not involving epithelial cells now, but mesenchymal or connective tissue. I hope we get that.
Yeah.
Only please repeat, sir. I didn't.
Hello, Dr. Olushola?
Yes, let me quickly repeat it, sir.
Okay, so what I'm saying is you got the aspect of metaplasia of connective tissue, what it means, Abi? You are having a change from one
From that
connective
side,
tissue.
from that part.
Okay, so metaplasia of connective tissue means that you're having a change from one connective tissue to another. For this myositis ossificans, you're having a change from muscle to bone. Muscle is a connective tissue. Bone is also a connective tissue. If somebody suffers fracture and there is muscle injury, instead of the stem cells of the muscle to differentiate into new muscle cells and replace the damaged muscle, some of them will not do that. Some of them will now differentiate. They will be reprogrammed. They will now differentiate along the pattern of forming bone to replace some of those muscle cells. So once you form bone on a muscle, you call that myositis ossificans. We don't actually know the reason why it happens. It just happens. It's not as if maybe it will help the muscle perform better or something. It just happens. So just notice hard mass within that muscle after healing. That is myositis ossificans. Formation of bone within a muscle after trauma due to differentiation along another mesenchymal tissue, along another connective tissue pathway. So from muscle to bone. Instead of it to be muscle to muscle, it's now going from muscle to bone due to reprogramming of the stem cells. The stem cells that are supposed to form the muscle are now reprogrammed to stem cells that now form bone. So bone is now being laid down on the muscle. That is myositis ossificans. It's a form of metaplastic change. So the last one, Dr. Olushola, are you clear now? Hello?
Yes, I'm clear. Thank you.
Okay. So the last one is dysplasia. So dysplasia is not actually considered a true adaptive response. It's just a fertile ground for malignancy to develop. It's a progression of hyperplasia or metaplasia. Hypertrophy doesn't go to dysplasia. It's only hyperplasia or metaplasia that can go to dysplasia. And when we say dysplasia, what are we talking about? Dysplasia is specific for epithelial cell only, not mesenchymal cells. It is only epithelial cells that can undergo dysplasia. And what does it mean? It means disorganized or disordered precancerous epithelial cell growth. That's what it means. So the epithelial cells are now proliferating, but they've not turned to cancer, but they are on their way of going to cancer. They usually
You said it's
follow
only what can turn to dysplasia.
in epithelial cells, only epithelial cells. It doesn't happen in
Oh,
connective tissue like muscle or bone, no,
okay.
or blood, no. You cannot have blood dysplasia. You can only have dysplasia of an epithelial cell, bronchopulmonary dysplasia, dysplasia of the, although that's not a very good example, though, but you can have cervical dysplasia. So when you say cervical dysplasia, you're talking about dysplasia affecting the epithelial cells lining the cervix. So what are the changes that we need to see as pathologists with respect to dysplasia? We are looking for two basic things. We are looking at the cell entirely. We are now looking at the nucleus. So what are you going to see in the cell? There's going to be loss of uniformity of the cell size and the shape. So let's look at what is happening here. This plastic one, we can see that the shapes are no longer uniform. You know, so they are irregular. We don't have uniformity of the shape again. Even the size, some are big, some are small. That's one thing that you see. Then because of that, there's going to be loss of tissue orientation. So the configuration of the whole tissue will be changed. Because we are seeing some tall structure, they are not like flat, like the ones we are seeing upwards. Everything is just scattered. The orientation has changed. That's another thing that happens. So the change in orientation and the change in the size and the shape are just like the same thing. So, but we have a specific term for the size and the shape that is changing. We call that pleomorphism. So non-uniformity of the size and the shape is called pleomorphism. So that is going to bring about a loss of tissue orientation. You know. Then at the end of the day, we'll now look at the nuclear change. So what happens at the nucleus? We are just going to notice that the nuclear cytoplasmic ratio is going to increase. There's going to be increase in the nuclear cytoplasmic ratio, and there's going to be chromatin clumping. So once you see all those things, that is. So pathologists usually have the normal values for the nuclear cytoplasmic ratio for each cell. So, but in a cell that is undergoing dysplasia, you notice that the ratio of the size of the nucleus through the cytoplasm is going to be more than normal. Then when you examine the nucleus in the microscope, you will notice clumping of the chromatin. There are so many things that can cause clumping of the chromatin, though, and we are going to talk about them. We can grade dysplasia. We can grade it. Grade 1, 2, 3. Mild, moderate, severe. Mild, moderate, severe. Now, the grading is based on all these changes that we have talked about. How is it progressing? That is, how is it moving from up to down? How is it moving from up to down? In the mild one, it's only affecting the cells that are, topmost. By the time it's getting to moderate, it has moved past the topmost cell, it has gotten to the middle layer. And by the time it's severe, it has gotten to the ones that are below. But it hasn't crossed the basal membrane of the cell. You guys can see this blue line. Can we see this blue line here?
Yeah.
Yeah, so that is the
Yes.
basal membrane. So once we have dysplastic changes in these epithelial cells, moving past the basal membrane, going to the next tissue, that is carcinoma, that is invasive carcinoma, right? That's like a malignancy. But in dysplasia, all these changes that we have talked about, the changes in the cell as a whole, loss of uniformity of the cell size, the cell shape, loss of tissue orientation, and the nuclear changes, which include increased nuclear cytoplasmic ratio and the clumping of the nucleus, the clumping of the chromatin. When it's happening at the cells that are topmost, that's grade 1. When it has gotten to the one in the middle, grade 2. when it has gotten to the one below, that's severe. That's grade 3. Now, that severe one is irreversible. Anybody that has severe dysplasia, we call that carcinoma in situ. It is irreversible. It is irreversible. So from the carcinoma in situ, it can cross the basement membrane and become invasive carcinoma. So it is usually preceded by a metaplasia or hyperplasia. So hyperplasia that is persistent. The stimulus that is causing that hyperplasia is not going. The stimulus that is causing that metaplasia is not going. So someone that continues smoking, the person can have cervical. That's why smoking is one of the risk factors for CA cervix. So if they continue smoking, they can have metaplasia, or no, don't let me use smoking. Let me say sexual intercourse. So with repeated sexual intercourse, there's increased trauma to that cervix. It undergoes metaplastic change. So it undergoes metaplastic change. From there, it can progress to dysplasia, then from dysplasia to cervical cancer. Or someone that is smoking repeatedly and the respiratory epithelial cells are undergoing metaplastic change. So they are changing from the pseudostratified squamous epithelium to stratified squamous epithelium. If that smoking stimulus doesn't stop, they will move from the metaplasia to dysplasia. So we are going to be seeing all these changes we have said in the cells. Then from there, they can now progress to cause squamous cell carcinoma of the lungs. So we must always remember persistent metaplasia and pathologic hyperplasia are potential causes of dysplasia which can lead to malignancy. So let's try to look at this question before we move. Okay, so a 62-year-old man is evaluated for worsening cough, shortness of breath, episodic hemoptysis over the past 6 months. The person is coughing, difficulty in breathing, and hemoptysis over the past 6 months. The patient has also had weight loss, 15 kg, over the same period without any change in his diet or activity level. He has a 50-pack year smoking history and does not use alcohol or illicit medication. The BMI is 18 kg per meter square. The patient appears cachectic with temporal wasting and generalized loss of muscle mass. X-ray shows a large lung mass with mediastinal lymphadenopathy and pleural effusion. Which of the following cellular process is most likely responsible for this patient's muscle loss? From this history, although I know we have not done it, what do we think is happening to this patient?
CA.
Yeah,
Lung CA.
please, let's be, just say it. Just say it. Don't worry if you are wrong or right. What did you say? Someone said it. Someone's already said the answer.
Lung CA.
Lung CA.
This is a lung cancer. Yeah. So this is lung cancer. So this person is having cancer and the person is having muscle loss. So they are now asking us, what is the mechanism of the muscle loss in this person? So what do we think will be responsible for the muscle loss in this person? What adaptation do we think is going to happen in this person? Is it hypertrophy? Is it hyperplasia? Is it atrophy? Is it
Atrophy.
Atrophy.
atrophy?
So A, ubiquitin.
Yeah. So it makes sense for us to pick covalent attachment of ubiquitin to muscle protein because of atrophy. So this guy is having malignancy. There's possibility of elaboration of the TNF that we've talked about, the interleukins that will activate the ubiquitin. And the ubiquitin will bind to those proteins in the cell and it will degrade those proteins, reducing the size of the cell. We also said that people that are having cancer, they have release of TNF. TNF is an interleukin, it's a cytokine that suppress appetite, so they don't eat well. So they have, reduced nutrition. Their protein synthesis reduces, and because of that, there's going to be reduction in the size of the cell. Okay, so let me ask us about the reprogramming of undifferentiated mesenchymal cells. So what is that talking about? What response is that?
Metaplasia.
Metaplasia. So metaplasia. We've not talked about the other ones. So let's just move. So we are going to talk about cell injury now. one thing about all these adaptive response is that it will get to a time that the adaptive response is going to become maladaptive, and the cell can no longer cope with whatever is happening to it, and they will die. But before they die, they'll get injured first. Let's use the example of ischemia. So somebody is having uh, rupture of an atherosclerotic plaque that is causing thrombosis of the coronary vessel. So the amount of blood that is delivered to the cardiac muscle is reduced. So that means that the cardiac muscles are suffering ischemic injury. Now, you guys remember that oxygen is essential for oxidative phosphorylation in the mitochondria. So
Yeah.
exactly. So if there's no oxygen getting to those tissues, that means that the mitochondrial activity will be paralyzed, and they will no longer be able to generate enough ATP. Hello?
Where with you?
Okay. And they will not be able to generate enough ATP through oxidative phosphorylation. ATP drops. Now, I want you guys to know that any form of injury that you want to explain, you will definitely link it to mitochondrial damage. If it's toxin, those toxins are going to damage the mitochondria. If it's radiation, they are going to damage the mitochondria. If it's ischemia, it's going to damage the mitochondria, and everything will culminate in drop in ATP. All cells of the body on their membrane, they have some pumps, particularly sodium potassium ATPase pump. I know that we are all familiar with this guy. Sodium potassium ATPase pump. What does it do? It is a pump that allows now it is ATP dependent, meaning it is moving electrolyte across their electrochemical gradient. So it's moving them against, rather not across, against their gradient. That is moving sodium to where it is abundant. If sodium is abundant in a place, if you want to bring sodium to that place, you need energy to drive the process. If sodium is moving down its concentration gradient, that is, it's moving from where it is much to where it's less, you don't need energy. But if you are moving sodium from where it is less to where it is more, you need energy to move it. Do you guys remember the cellularity of sodium? Is it extracellular or intracellular? Anybody?
Extracellular.
Sodium is extracellular. Now, what the sodium potassium ATPase pump is now doing now is that it's now pumping intracellular sodium from the cell into the extracellular space. That means that it needs ATP to do that. And it's now moving that, and it's moving 3 molecules of sodium from inwards to outward. It's moving potassium from where it's merged intracellularly, from where it's merged, which is sorry about that. It's moving potassium from the outside to the inside. The potassium is more in the inside. It's now moving potassium from the outside where it's few into the inside where it's reduced. Where it's more, you need ATP to do that. So the sodium potassium ATPase pump is a pump that moves sodium from the intracellular compartment where it is little to the extracellular compartment where it is much, and it moves potassium from the extracellular compartment where it is little to the intracellular compartment where it is much. You need ATP to drive that process. So once ATP is reduced, that means that you cannot achieve that movement. It means that all the sodium will be pulling where will the sodium be pulling? Anybody? If you cannot
Into the intracellular.
yes,
Intracellular.
so sodium will be intracellular. Sodium will stay intracellularly. Now let us stop there. Same thing will happen to potassium. Potassium will be pulling extracellularly, but it is that sodium that is pulling intracellularly that we are interested in, because sodium is osmotically active. Sodium will now draw water into itself. When it is intracellular, it will draw water into itself. So water wants to stay with the sodium. So in the process, the cells are going to swell. They are going to engorge. Now, there will be an increase in the intracellular concentration of calcium too. Why? We have done cardiology, so although some of them have not done it, so I'll try to explain. The reason why calcium will accumulate in the cell is that there is another pump on the membrane that we call the sodium calcium exchanger. So what that one is always there to do is just to exchange sodium for calcium. so that it can increase the intracellular concentration of sodium. So whenever the cell notices that sodium is low, what they will do is that they will take calcium out so that they can bring sodium in from extracellular into the intracellular. Now, in someone that is having reduced ATP and sodium cannot be moved out from intracellular to extracellular, it means that the sodium that is intracellular is high. That means there's little need to exchange sodium for calcium. So calcium doesn't go out, calcium will stay inside. So there's reduced activity of the calcium sodium exchanger. So the amount of calcium intracellularly increases. All these things is going to culminate in cellular swelling. Sodium is going to attract water, the cell will swell, and what will happen? And this cellular swelling is the earliest morphologic manifestation of reversible cell injury. Please, we must always remember that. Reversible cell injury, the first thing that happens is this cellular swelling.
Cellular swelling.
Yes. So, what will now be the consequence of this cellular swelling that is happening? All the other organelles, they will now start misbehaving. They start misbehaving. And in response to this reduced ATP, the cell will want to, generate energy via the anaerobic pathway. The anaerobic pathway is going to increase the production of lactic acid. We remember that. So when lactic acid pools within the cell, it decreases the pH and is going to cause chromatin clumping. So what is responsible for clumping of the chromatin is actually due to the lactic acidosis. So we can remember that immediately. So when the ATP drops and this pumping stuff happens and the cellular swelling, the next thing is the lactic acid starts accumulating in the nucleus and cause chromatin clumping. As the swelling occurs, it's going to destroy the cytoskeleton within the cell membrane, and it will make them form what we call blebs. So blebs are just like outpouching of the cell membrane, like hydropic swelling of the cell membrane, forming blebs. Can we see this? So it's just like the water is filling into those cell membrane. You can see this. So that's another consequence of the cellular swelling. They immediately start forming membrane. Mind you, the integrity of this membrane is not compromised. The membrane still remains intact because the intact nature of that membrane is very, very significant. In cellular swelling, in reversible cellular injury, the membrane is intact. We have to always remember that. The only thing that is happening is just blebbing. So hydropic swelling of those membranes. They just form some outpouching. We've said that the nucleus will clump due to the lactic acidosis. Now, what will happen to the endoplasmic reticulum? Endoplasmic reticulum will start losing its ribosomes. It will start losing its ribosomes. So when it loses its ribosome, there's going to be decrease in protein synthesis. There's going to be decrease in it. You guys forget this polysoma. Polysoma just means aggregation of ribosomes. So that's what it means. So the ribosomes are going to detach within the endoplasmic reticulum, and there's going to be a drop in protein synthesis. All these things will culminate in loss of function of that cell. Like the cell we are initially talking about is the myocardiac cell. So just few minutes, like 1 to 2 minutes after that ischemia, the myocardial cells can no longer contract again, and the heart will start failing. There's also something called the Myelin figures. So these Myelin figures are actually due to release of some free radicals. All these things will cause the release of free radicals that will now attack the lipids of this cell membrane. So they kind of react with the lipids. So we'll talk more about peroxidation as we are moving deep into this lecture. So those free radicals will react with the lipids in the cell membrane, and they will form a peroxidized form of the lipids. But the cell membrane that actually being affected are the membrane of the organelles, not the cell entirely. It's more of the membranes of the organelles, like membranes of the endoplasmic reticulum, membrane of the mitochondrion. So they are going to aggregate together and form Myelin figures. So it's one thing that we must always remember. So the first thing that happens, the ATP drops. When ATP drops, it's going to affect the sodium potassium ATPase pump. There's an increase in the intracellular concentration of sodium and calcium. Water pools in the cell, and the cellular swelling. When there's cellular swelling, what happens? The plasma membrane of the entire cell starts forming outpouching. hydrolytic vacuolization of those membrane, leading to formation of blebs. The cellular swelling is going to separate and pull out ribosome from the endoplasmic reticulum. Protein synthesis is going to drop because of the fact that the cells are now diverting towards the anaerobic pathway that is not involving the mitochondrion to generate energy in form of ATP. They are going to end up releasing lactic acid. The acidic nature of that molecule will now cause clumping of the chromatin. Release of some free radicals will interact with the lipid part of the membrane, causing peroxidation of the lipid of the membrane of the endoplasmic reticulum and the mitochondrion. When they are when the lipid within their membrane becomes peroxidized, they aggregate together and they form myelin figures.
Hello, Dr. Bali.
Yes, Dr. Zulia.
Yes, I have a question. The part of the decreased activity of the calcium, like I'm trying to understand that part. Like, what is
No,
the calcium supposed to do before?
yes, I get you. See, what they are saying is not decreased activity of intracellular calcium. They actually meant calcium pumps, sodium calcium pumps. Do you get?
Okay.
There's something we call
Okay.
the sodium calcium exchanger. Are you there? Now,
Yeah.
that sodium calcium exchanger is there so that you can maintain intracellular amount of calcium. So that whenever intracellular amount of sodium rather, so that whenever there's a fall in the intracellular concentration of sodium, you rapidly exchange it for calcium. So you bring sodium from the ECF into the ICF, and you bring calcium from the ICF to the ECF. You exchange it. Are you following?
Yeah.
Now,
Yeah.
what happens when the sodium potassium ATPase falls? What happens to the intracellular concentration of sodium? Does it rise or reduces?
Intracellular concentration of calcium will
Intracellular will
increase.
increase.
Of sodium.
It will increase.
It will increase.
Increase.
It will increase. So if it increases, do you think there's a need for that sodium calcium exchanging?
No, not
No,
anymore.
exactly. So there's a reduced activity of the sodium calcium exchanger. That is
Okay.
it. So
Okay.
intracellular
All
calcium
right.
will increase. Yeah. So that's what happens. Now, if the insult, you know that where we have stopped now is myocardial ischemia. We have not even got into myocardial infarction because they are
Mm-hmm.
different things. Now, if that insult continues and there's no establishment of perfusion again, that occlusion is still there. Then MI now sets in. So that's when we now start talking about STEMI and our non-STEMI. So
Non-steroid.
all these unstable angina, all these what's it called? Stable angina, we are still talking about ischemia. It has not
Skin.
caused death
Okay.
of the cell. It's just about to die. It's still undergoing reversible injury. By the time it gets prolonged and the cell cannot cope again, they will die by irreversible cell injury. And there's nothing you can do at that point. Now, um, in short, we can say that irreversible cell injury is a form of necrosis. It is a form of necrosis. So when the insult continues, what will now happen eventually? We remember that we have said that some changes happen to the cell membrane. They form blebs. Now, it will get to a time that this blebbing will be so severe that it will now eventually break down that plasma membrane. Once it breaks down the plasma membrane of the cell, cellular contents are going to be released. That is why cytosolic enzymes will be released in a myocardial cell that has suffered prolonged ischemia, prolonged hypoperfusion. So that is why one of the markers for MI is what? Troponin what? Troponin. Do you guys get? Because in someone that has ischemia, you don't need to look for troponin because you will not find it because the membrane is still intact. But someone that has suffered MI and there's death, there's irreversible injury of the myocardial cell, there's compromise of the plasma membrane, cytosolic enzymes have leaked out. Example is troponin. So it makes sense for us to now start looking for troponin. So if you see elevated troponin, we can say that this person is having an MI, not a myocardial infarction, and not ischemia. Do you guys get what we are saying now?
Yeah.
Yes.
Okay. I know that people that have done cardiology might be able to relate that with that very well.
Yeah.
So let's just stick with the fact that okay, in irreversible form of injury, the reversible form is not resolved. So the insult continues. So what will now happen? The blebbing is now going to cause damage to the membrane. So when the membrane of the cell damages, that means that cellular component can escape. Part of what escape includes the cytosolic enzymes, lactate dehydrogenase, creatinine kinase, troponin, depending on the particular cell that you are looking at. This one now is because we are looking at a myocardial cell. That's why they are giving example of troponin. So all those enzymes, they leak out of the cell. Calcium is the bad guy. Calcium will now find its way into the cell, which is bad. You don't want to allow intracellular calcium in the cell. It's very bad because calcium will activate some series of enzymes, including calcium-dependent phospholipase and protease enzymes. When those protease enzymes are activated, they will now start finishing the cell completely. They will go to all the mitochondria, destroy all of them. There's complete cessation of electron transport chain, ATP drops. They will destroy the membrane of the lysosomes. Lysosomes themselves, they contain hydrolytic enzyme. They will elaborate the hydrolytic enzyme into the cytoplasm. That will destroy the cell too. Autolysis, self-destruction. The nucleus is going to undergo degradation, but mind you, the changes that you see from this nuclear stuff is not actually due to the elaboration of the enzyme. It occurs independently of those guys. So what are these changes? There are three. Pekinosis, karyoexis, and karyolysis. Guys, once you see this, you know that that cell it has died. That is no longer reversible. You can see this in both necrosis and apoptosis. But apoptosis will not typically have this karyolysis, which is the last one. But what is common to both of them is pekinosis and karyoexis. Don't forget, we have said that irreversible cell injury are changes that you see in necrosis. True, they are like the same thing. So what are the nuclear changes? There's nuclear degradation, and it undergoes some changes. It's like a progression. Number one is pekinosis. So the nucleus is going to condense. The chromatin in the nucleus will first condense. It will condense. It will condense. When it condenses, it kind of shrinks the cell a little. The next thing is elaboration of some endonuclease. So those endonuclease will want to degrade all the chromatin fragment. They want to degrade the DNA and the RNA. So they cause nuclear fragmentation. So you notice that the chromatin are irregularly scattered within the cytoplasm. The first thing is pekinosis, condensation. It will condense. That's the first thing. Now you now want to break down those condensed chromatin into fragments. You employ the endonuclease enzyme. Endonuclease will break down the DNA. You need endonuclease to break down DNA and RNA. So they will break down the DNA in the chromatin. So you have chromatin fragmentation. So you see them irregularly scattered in the cytoplasm. At the end of the day, you just notice that there's slices of the nuclear membrane and the entire nucleus disappears. You don't see it again. That's cryolysis. 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 So that is that. And there's also something called amorphous densities or inclusion. These are typically seen in the mitochondrion. So these densities are seen in the mitochondrion that are still persisting because it's more like an aggregate of the lipid membrane of this mitochondrion chelating calcium. So all the calcium that have entered into the cell, they will chelate with those membranes of the mitochondrion and they will deposit within the mitochondrion and they will appear as amorphous density. If you don't remember this, it's fine. But most importantly, you must remember the breakdown of the plasma membrane, the nuclear degradation pattern, and the rupture of the lysosome. Those are the clinchers for irreversible cell injury. Very important. When the cell finally breaks down, so when they tell us what is the hallmark of irreversible cell injury, number one, the starting thing is the membrane breaks down. When there's a compromise of the membrane, all the things within the cytosol will leak out. Everything that is there, they will leak out, including the enzyme, the protein, everything goes out. That even has a relevance because inflammatory response is going to come. That is why necrosis causes inflammation. Apoptosis doesn't cause inflammation. We are going to get there. Because in apoptosis, the membrane is intact. Just like in someone that has reversible cell injury. So because of the damage of the plasma membrane in irreversible cell injury, the cellular content leaks out. The cytosolic enzymes are outside. calcium will be able to find its way in. You guys remember that calcium is an extracellular um, element. It moves from the extracellular space into the cell when there's an opening. So once it gets there, it starts activating protease and phospholipase enzymes. They are degradative enzymes. Those guys will now start cleaving the lysosome, the mitochondrion, particularly. So when there's a damage to the mitochondrion, there's going to be reduction in ETC and ATP level will fall. It damages the lysosomes, there's autolysis. Because lysosomes will also elaborate their own hydrolytic enzymes, which will cause self-destruction in form of autolysis. Then what are the nuclear changes? The three: pekinosis, cryoexis, and cryolysis. This is different from the nuclear change in reversible. In reversible, it's just nuclear clumping. Membrane is not ruptured. It's just forming bleb. That is it. But here, membrane have ruptured, things have entered, calcium is there, releasing protease and phospholipase degrade the cell, and there are some nuclear changes. What are those changes? Pekinosis, cryoexis, and cryolysis. So in pekinosis, condensation of the chromatin. Cryoexis, splitting of the chromatin into fragments. Then cryolysis is now eventual disappearance of the nucleus totally. That's irreversible cell injury. Is it clear to us? Or do we have any question?
It's clear.
Okay. So, um, let's start talking about the pattern of cell death. So we've used time to talk about necrosis, which is irreversible cell death. Apoptosis has its own specialized pattern of death, how it happens. But one thing about apoptosis is that it is ATP dependent programmed cell death. That is, it
Dr.
occurs in
Babadi,
cells. Yeah, Dr. Olushola.
I wanted to add that, you know, is it, is it, could it be true that, the first one we mentioned was necrosis, right?
Okay.
So in the process of going through that necrosis to the end, pata like the end of us having karyolysis,
Okay.
did it ever pass through the process of probably mitochondria, membrane damage, cytochrome C, and then it progressed? Or it's a different pathway entirely, necrosis and apoptosis?
Yeah, so when you are talking about the cytochrome C, you are already talking about apoptosis, which is a different pathway. Do you get, Dr.
Okay. Oh,
Olushola?
all right.
That is a
Yeah, okay.
separate pathway. So cytochrome C is not involved. We are going to see a question like that, so we'll be able to relate. So apoptosis is ATP dependent programmed cell death. It typically occurs in cells that the body have labeled that they will die. As the body is growing, it knows some cells that this one where they see, this one will die. It's not just yet time. When it's time for it to die, it will die by apoptosis. But there's an exception. It also occurs in cells that you have not really programmed, but because of some insult that has happened to those cells, they will die by apoptosis. Let's talk about the pathways for apoptosis. Three part is before it's usually two, but now they've separated into three. So all the stuff they are evolving, stuff are getting manufactured every day. We have three pathways now: intrinsic, extrinsic, and the perforin granzyme B pathway. I know we are familiar with this perforin granzyme. Which cell releases perforin granzyme? Anybody?
NK cell.
Cytotoxic T-cell.
Cytotoxic,
NK cell, natural killer cell.
you guys are fantastic. The stuff is there. Perforin granzyme B is released by typically NK cells and the cytotoxic cells. Those three pathways culminate in the activation of caspases. That is it. Those caspases are called cytosolic proteins. That is, proteins that are involved in destroying cytosolic components. They just destroy the cytosol. That's what they are for. Caspases. Those three pathways, that's what they culminate in. They are just, different pathways through activation of caspases. That's what they want to do. Before I get to what will happen, let's talk about those pathways individually before we come back. So let's start with the intrinsic mitochondrial pathway. So we've said that apoptosis is a programmed cell death and is ATP dependent. And we said that it typically occurs in cells that have been predestined to die. One of that example of predestined death is remodeling in embryogenesis. You guys will agree with me that it is not all the cells that start the process of embryogenesis that will end up with us. Have been our doctors really at? Is that so? Is it all the cells that we start during our embryonic phase that will end up with us as adults? Is
No.
it? No. Some of them will die. What is the pathway in which they are going to die? Apoptosis. Apoptosis. Because that's a predestined death. Let me give you an example. For example, in CNS development, some part of the notochord are going to regress and they will die off. The remaining will persist as the nucleus pulpose. Another one is thyroglossal duct that connect the back of the tongue to the thyroid gland. Once the thyroid gland gets to its usual position in the neck, that duct will disappear. It will obliterate it will obliterate and die off by apoptosis. But in some individuals, it can persist, but that is an abnormality. Typically, it's supposed to regress and die by apoptosis. So that means that we can say that apoptosis is involved in embryogenesis. Another form of apoptosis, again, is death of cell when the regulatory factor that is stimulating the proliferation of those cells are withdrawn. For example, somebody have a bacterial infection. In response to those bacterial infections, we want to release interleukins that will help us produce T helper cells, that will help us produce bacteria to respond to that, that will help us produce macrophages that will respond to that bacteria and they will kill it. They will kill it off. At the end of that immune response, when they have killed the bacteria, what will happen to the level of interleukin that is stimulating it? It will drop. We don't want those T cells and the macrophages to be existing again because we don't need their services. They are done. They've done well, but they need to go. How will they go? They will go by apoptosis. So that statement is very important. Whenever a regulatory factor is withdrawn, the proliferating cell population will die off by apoptosis. Now, that intrinsic pathway is also called the mitochondrial pathway. I will tell us why. And this third reason why apoptosis is happening is more relevant to that intrinsic name, because the third reason why apoptosis can be triggered through the intrinsic pathway is when there's an insult to the DNA within the cell. So that's an intrinsic problem. There's an intrinsic problem. I've insulted those DNA, you have damaged them, either maybe due to radiation exposure, you've broken the DNA strand, or there's toxin that is damaging the DNA. Those abnormal DNA are going to activate something that we call P53. So P53 is now going to go through some other pathways to trigger apoptosis. Now, what I just want to emphasize here is that these three things that we have said activate the intrinsic pathway or the mitochondrial pathway of apoptosis. Number one, embryogenesis. Number two, when a regulatory factor is withdrawn. Number three, insult to the DNA, either from radiation exposure, from infection, from toxin, from hypoxia, anything. They will activate the intrinsic pathway of apoptosis. Now, this intrinsic pathway is also known as the mitochondrial pathway. It's very, very important to remember that, because there's going to be something that will be escaping from the mitochondria that will trigger the apoptosis. You guys remember that we've said that all the three pathways, they culminate in the activation of caspases. Caspase is what we call the ventral apoptosis. So how will the mitochondrial pathway now activate its own caspase? Let's take the DNA injury for an example. So when there's DNA insult, either from radiation, that abnormal DNA needs to be taken out. It should not be there, so that tumor or malignancy will not happen. That is why we have our tumor suppressor gene like TP53 available to protect us. So that damaged DNA will activate the P53. What P53 do? P53 will activate some group of protein that we call the bad protein. Backs and back. Backs and back. These backs and back protein, they are pro-apoptosis. They want to kill off any cell that is labeled to die through apoptosis. Where do they stay? They stay within the outer membrane of the mitochondrion. They're already there. They are there. Alongside with another family of protein that we call BCL2 and BCLXL. But those ones are anti-apoptosis. When there's DNA insult and there's activation of P53, P53 will activate this back and this backs protein. When they get activated, they are going to make pores within the membrane of the mitochondrion, and they are going to deactivate the activity of BCL2 and BCLXL. When they do that, caspase is going to, cytochrome C is going to escape from the inner mitochondrial membrane across those pores into the cytoplasm of the cell. When the cytochrome C gets into the cytoplasm of the cell, it will combine with some factors. I don't want to mention that because their names are complex and I don't think we need to remember them. But the only one I want us to remember there is ATP. It will combine with ATP alongside with some other protein that will activate caspase. They will activate caspase from the initiator caspase to the executioner caspase. So when let's know that, okay, when the cytochrome C binds to ATP, it will activate caspase. When caspase gets activated, something is going to happen. Let us keep that for now. It's going to now trigger the remaining apoptotic process, which we are going to talk about. So that is the mechanism how the intrinsic pathway is activated. So in someone that is undergoing the process of embryogenesis, in somebody that wants to complete and stop immune response, in somebody that has suffered damage to the DNA from radiation, from hypoxia, from infection, and they want to kill off those cells, what they will do is to activate P53, all of them. When P53 is activated, P53 will activate the backs and the back so that they can puncture the mitochondrion. When they puncture the mitochondrion, they will have deactivated the BCL2 and the BCLXL. Those ones are there to protect the integrity of those membranes. So their whole activity is compromised. They are not working. So backs and back take over because they're activated. They make pores. Cytochrome C finds its way from the inner membrane of the mitochondrion across those pores into the cytoplasm. When they get to the cytoplasm, they bind to ATP in the cytoplasm and you activate caspase. What is the clinical relevance of some of these proteins? Let's look at the BCL2, for example. We've said that this BCL2.
Please, before you move on, the TP53 part of it, like I kind of like didn't get that because I was thinking of how TP53 works and I missed that explanation you said.
Like we are not going to go to the way it works by stopping G1 to GSO. You know that TP53 is a tumor suppressor. You remember that, Dr.
There's a tumor suppressor, yes.
Now, a DNA that has suffered damage, what do you think should happen to that DNA? Should it be damaged or should it be kept in the cell?
It should be damaged.
It should be damaged. It should be removed. How will they remove it? So, and if you are removing a damaged DNA, that means you are suppressing the occurrence of tumor, right?
Yeah.
So that means that it's reasonable to say that we are employing the services of a tumor suppressor gene.
Okay.
So that's why DNA damage will now activate a tumor suppressor gene, which is what? P53.
Okay.
When P53 is now activated, what will happen? There will be activation of the back and the backs. What will they do? They will make pores in the mitochondrial membrane. What will these pores do? These pores will now allow cytochrome C to move from the inner membrane across the pores into the cytoplasm. When the cytochrome C gets into the cytoplasm, it's going to combine with ATP so that it can be charged to activate caspases. So caspase is now what will now destroy those DNA that we are talking about. So cells that are having damaged DNA, it will take them out. Well, we are going to explain how later. Let's just keep that one Now, do you get that,
No, thank
Dr.
you. Yeah,
Zubin?
I do. I do.
All
Thank
right.
you.
So let's continue. BCL2 is one of those anti-apoptotic protein. It binds to the membrane of the mitochondrion so that the cytochrome C doesn't escape and be activating caspase that will now cause apoptosis. So they are anti-apoptotic because they don't want caspase to be activated. They don't want cytochrome C to leave and activate the caspase. It means that any cell that has mutation involving BCL2, it means that the cell will not die. Abi now, guys, if a cell has mutation that's affecting BCL2, that BCL2 is overexpressed and it will not allow apoptosis to occur because it will not allow cytochrome C to move out. That means that it will
Yeah.
not activate caspase. That means that cell will not die. Abi now,
Yes, it will
it will not
remain.
die.
Yeah.
It will not die. So that is the pathophysiology of some lymphomas, particularly follicular lymphoma. Follicular lymphoma is associated with translocation of chromosome 14 to chromosome 18. Chromosome 18 codes for the BCL2 gene. The BCL2 gene is located around a locus within chromosome 18. So when there's this translocation with chromosome 14 encodes the heavy chain of the immunoglobulin. The heavy chain of immunoglobulin, the gene is encoded within chromosome 14. Once there's translocation of chromosome 14 with 18, once that fusion occurs, there's constitutive activation or constitutive expression of BCL2 protein. It will just be expressed continuously, uncontrolled. And it has fused with the cell that is containing this mutation. And let's say this cell now is now the B cells because it is B cells that will have this mutation. It is B cells that needs to be producing long line of hemoglobin. Abi now, I don't know if you guys are able to follow what I'm saying. It is the B cells that needs to be producing immunoglobulin, right?
Yes.
So it means that if the chromosome 18 within the B cells are fused with chromosome 14 and there is uncontrolled expression of BCL2, it means that you will not have death of those cells. Those
Yeah.
cells will continue to express that protein that they are expressing, which is the immunoglobulin. So you are going to have continuous expression of immunoglobulin. You are going to have continuous proliferation of the B cells as well,
B cells.
because of the anti-apoptotic action.
18.
Exactly, because of the anti-apoptotic action of the BCL2 that is
18.
overexpressed. And how is that happening? BCL2 will block the mitochondrial pulse so that cytochrome C cannot escape. There's no cytochrome C to activate the caspase. There's no ability to drive apoptosis. So that means that the cells will not die. They will continue to exist for life. That's the basis of follicular carcinoma. Don't forget.
So it's the long arm that it affects?
Yeah, the heavy chain. The heavy chain.
So what happens to the short one?
No. This chromosome 14 just encodes for that heavy chain. Do you understand?
Okay.
So in that process, it's going to affect the entire cell. That is it. So,
okay.
So that is that about the intrinsic pathway. Let's go to the extrinsic pathway, the death receptor pathway. There are two receptors in this death receptor pathway: the TNF alpha receptor and the FAS protein. This FAS protein is also a receptor. Both of them are structurally similar. So this FAS protein or FAS receptor is belonging to the family of TNF alpha. Now, they have receptors. There's a FAS receptor. There's a FAS, which is the FAS receptor. There's a TNF alpha receptor. They have ligands. The ligand that binds to FAS, because FAS is like a TNF receptor, is FAS ligand. The ligand that binds to TNF receptor is what? TNF alpha. Let me take that again. In the extrinsic pathway or the death receptor pathway, it's involving activity of two receptors: the TNF alpha receptor and the FAS protein. So FAS protein is a receptor, and both of them have ligands. We have the FAS ligand, we have the TNF alpha, which is a ligand that is binding to the TNF alpha receptor. Interaction between these receptors, so the interaction between FAS ligand and FAS an interaction between TNF alpha and TNF alpha receptor can activate apoptosis. It can also activate caspases. So once you have fusion of those guys together, they will activate the caspase directly. I want to say that the TNF alpha receptor and the Fas protein is present in almost all the cells in the body. It's present in almost cells of the body. The TNF alpha receptor interaction is usually very important in the elimination of some tumor cells. So if you want to destroy tumor cell as a cytotoxic cell, if you know that part of the cells that respond to tumor cells are what? Cytotoxic T cells. So what they will just do is that the TNF alpha that is expressed from the cytotoxic T cell will just bind to the TNF alpha receptor on that cancer cell. And once there's a fusion, that fusion is going to activate the caspase that will trigger apoptosis of that cancer cell. That's the importance of the TNF alpha component. But the Fas component is important in thymic medullary negative selection. I think you guys will agree with me that this kind of death is also a destiny death. It's not all the T lymphocyte that find its way into the thymic medullary area will survive. We remember that from our immunology that we just concluded. Babi, negative selection.
Is it that AIRE something?
Yes, AIRE. Please, I hope we have not forgotten this one that we are not answering.
Yeah, we remember.
Okay. Some of those T lymphocytes within the medullary area of the thymus will be deleted. How will they get deleted? They are getting deleted by apoptosis. What pathway of apoptosis? The extrinsic pathway. Specifically, activation of the Fas pathway. So what happens is that, like I said, Fas receptor, that's like repetition because Fas is like a receptor itself. You guys are already following. You get what I'm saying. So that Fas.
Please, Dr. Warden, sorry, please. Before you go far, you were saying something about small necrotic factor, but I lost you for a second.
Yeah. So tumor necrotic factor, I said it is elaborated from the cytotoxic T cell. And that tumor necrotic factor receptor is present in some cancer cells. So if cytotoxic T cell want to eliminate some cancer cells, you know that cytotoxic T cell have anti-cancer activity. You know that. So if they want to destroy those tumor cells, they can bind to the TNF receptor on the tumor cell. So when they bind to the TNF receptor with their own TNF alpha, they are going to activate the caspase that will now trigger apoptosis of those cancer cells. Do you get it?
Yes, I get it. Okay, all these infliximab, all these are the work. Okay.
Yeah. So infliximab is another thing. If you want to now modulate immune response, you can use infliximab. So now we are now talking about the Fas interaction. The Fas interaction is in a different setting. Fas interaction is helpful in the deletion of self-reacting T lymphocytes. That is T lymphocytes that are present within the thymic medulla that are reacting to self-antigen. So once you elaborate the AIRE, which is the autoregulatory molecule that is presenting tissue-restricted self-antigen to the T lymphocyte, when they see it and they are responding to them, if those T lymphocytes are responding to them, they know that these ones are self-reacting T lymphocytes. They will take them off and kill them. How will they kill them? They will kill them through this extrinsic pathway by using the Fas interaction. So like I said, Fas is a receptor. It is present in all cells. Fas ligand is predominantly expressed by the cytotoxic T cell. So once those cytotoxic T cells identify those self-reacting T cells, the cytotoxic T cell will now use its own Fas ligand to bind to the Fas on those self-reacting T cells so that they can delete them by apoptosis. Because that interaction is also going to activate the caspase that will take those cells out by apoptosis.
So, Dr. Babale, so that reaction is between T cell and T cell.
Yes, fantastic. It is between T cell and T cell. It is between cytotoxic T cell and self-reacting T cell. So you can induce negative selection in the medullary aspect of the thymus. There's a clinical significance to that.
But the one up there,
Hello?
Dr. Bal,
Yes,
sorry
Dr.
to
Precious.
cut you. Sorry to cut you. Dr. Zulia said this the fast and fast ligand is between cytotoxic T cell and the under T cell in the hair stuff, negative selection. But that means this tumor necrotic factor alpha is between a T cell and a tumor cell.
Yes, you get it. It's between a T cell and a tumor cell. So the one that is bearing the receptor is the cancer.
Okay.
The one that has the TNF alpha is the cytotoxic T cell.
Yes.
So
Yes.
let's say that.
Dr. Babale.
Yes, Dr. Olushola.
So is all cells expressing the TNFR2?
Yeah, all cells express it too. All cells express
Okay,
it.
so what now makes them to this process of binding to? What triggers it?
Yeah, so that one I cannot explain. I don't have an explanation for that. But I know that the TNF is expressed in almost all the cells of the body. So I don't want to say that it is predominantly expressed in cancer cell. That would be wrong. But I don't have an explanation for that. So maybe the T cells just have a way they are able to, the thing is, there's a possibility that what will be shown on the surface of the tumor cells will differ in terms of this antigen presentation. We're talking about this
Antigen presentation.
CD8 expression of a thing. So it's possible that there will be down regulation of that CD8 receptor, because natural killer cells, cytotoxic T cells, they try to eliminate any cell that is not normal. Because they expect all the cells to be expressing those CD8 receptor within. Sorry, I said CD8, MHC, one receptor within their surface. Do you guys get that? So there will be something that the cytotoxic T cell will actually use to recognize it, that this is a tumor cell. Although that one is beyond the scope of this. Sure you get. So, but I know that there'll be something that they will use to, okay, this is actually a tumor cell. Maybe by down regulation of the MET, or maybe just by presentation, by antigen presenting cell that's containing, maybe, okay, antigen presenting cell that contains the MET1 now, binds to some of those tumor antigens. They're able to present them to CD8 cells, and from there they go to attack that cancer cell. I don't know if you guys get what I'm saying. You know that our cancer cells can be recognized by some of those cells. So it's possible that they have been attached to antigen presenting cells that will now take them to the cytotoxic T cell for destruction. So that is that. So
hello, Dr. Bala.
Yeah, Dr.
Hello.
Dr. David.
Okay. Yeah. So you said that this fast and fast ligand interaction is between the cytotoxic T cell and the self-reacting T cell. I was thinking that this negative selection thing is between the T cell and the self-antigen. Like the T cells are trying to recognize the self-antigen so they don't get to destroy.
No. Negative selection means that you want to take out. You want to negatively select self-reacting T cells. That's what you are doing. Like, what you are doing initially is that you will employ the activity of AIRE. That AIRE is like a protein that is containing all the self-antigens in the body. You know that all the self-antigens in the body needs to be shown in this thymus, in this medullary area. How will you do it? It is AIRE that is containing all the copies of the cell antigen in the body. Do you get it, Dr. David? Hello?
Right.
So that AIRE now is like containing, the word was tissue restricted self-antigen. That is, all the self-antigen that is native to each tissue, they will now bring it to the thymic, to the medullary area of the thymus. When they now show it to those T cells that are there. Anyone that now reacts to those antigens, it will be labeled as a self-reacting T cell. So the next thing is now to negatively select it out. We want to delete it from the population. So that is where this FASL interaction now comes in. Do you get it, Doctor?
Yes.
So that is
Thank you
the
very
way
much.
it works. So now there's a possibility of failure of the expression of this FAS receptor, and if that happens, you cannot have this FASL interaction. So if there's a defective interaction, a condition can ensue from that, and that is the autoimmune lymphoproliferative syndrome. So all the T cells that are supposed to be negatively selected, they are not. So that means that the numbers of self-reacting lymphocytes will increase. Now these guys will just be proliferating in almost all the lymphoid organs. They go to the lymph node, they'll proliferate there, they'll cause lymphadenopathy, they go to the spleen, they cause splenomegaly, they go to the liver, hepatomegaly. And because of the fact that they are self-reacting, they can destroy self-antigen. They have predilection for destroying the cells in the bone marrow first. Although they can affect other tissue, they can cause so many autoimmune diseases. But I think for the scope of our step one, we should just focus with autoimmune cytopenias and close our eyes to the other one because there's a lot of them. So they first proliferate in the lymph node, they cause lymphadenopathy, they proliferate in the liver, hepatomegaly, the spleen, splenomegaly, and they start attacking self-antigen, particularly those that are found within the bone marrow. And in the process, it's going to cause autoimmune hemolytic anemia, it's going to cause autoimmune neutropenia or lymphopenia, and autoimmune thrombocytopenia. So the constellation of those three will bring about autoimmune cytopenia. So I think that should make sense to us. It's just like failure of this negative elimination. So if there's failure of this guy, it will increase the number of self-reacting lymphocytes, and you can have lymphadenopathy, hepatosplenomegaly, and autoimmune cytopenia. Do you understand this?
Yes.
All right. So,
Mm-hmm.
so the last pathway is the perforin-Granzyme pathway. This one is very straightforward. Very straightforward. So what happens is that cells that release perforin and granzymes are the ones that are involved in this pathway. What are these cells? The natural killer cells and the cytotoxic T cells. So what would they do? Whenever they bind to any cell that they want to kill, let's say they bind to one tumor cell or they bind to any cell or virus, if they want to kill it, what would they do? They will release this perforin first. So what will perforin do? Perforin will make hole in that cell. So I think we have an image. Yeah. So perforin will make hole in that cell that they want to damage. When it makes hole, it will allow granzyme to be able to enter the cell through that hole. And granzyme B is just like cytochrome C from the intrinsic pathway. Granzyme B, just like cytochrome C, activate the executional capsids. So they can trigger apoptosis. We can see that one is very straightforward. That one is the easiest to. Once it fuses with the target cell, they will release perforin. Perforin will make a hole in the cell. Granzyme B will be able to enter. When granzyme B enters, it now activate the caspase and now drives the apoptotic process. So let's now go back. We've talked about the pathways. Let's go back to what happens. So when there's now activation of the caspase, what happens? What happens now? You can see what is happening here. Two things essentially. Number one, the caspase is going to come around here and it will cause condensation of the nucleus. So it release some this once the caspase is activated, it will cause condensation of the nucleus. When the nucleus condenses, the cell will shrink. The cell shrinks. The caspase at the same time will also, because it's like a cytoplasmic protease, it will destroy the cytoskeleton of the cell membrane. So in the process, it's going to form cytoplasmic blebs. Now, this bleb is different from the bleb of reversible injury. What this bleb will do is that they are going to take out portion of the cell. They're just going to take a small portion that is containing some mitochondrion, some nuclear material, some endoplasmic reticulum, some lysosome. They will form some bodies. They will move out. Another one will be formed like that. They will move out. They will form. So that those bodies will be recognized by macrophages. They are like pick me, eat me signals. So once they come out of those cells into fragments like that, they will be recognized by the macrophages, and the macrophages will take them out. There's never a compromise of the cell membrane in apoptosis, and there's no inflammatory response. This is an adaptive mechanism to ensure that there's no inflammatory response to apoptosis, so that the cellular content doesn't leak out. They will just form, they will engulf them into these blebs. So the blebs will travel out as this apoptotic body, and they'll be recognized by the macrophages, which will now digest them. So some other nuclear change that will happen is pekinosis. There's going to be pekinosis. There's going to be cryolysis. There's going to be pekinosis. The chromatin will condense. Then subsequently there will be fragmentation of the chromatin, but you don't typically have this nuclear cryolysis. It's not typically there in apoptosis. And one thing I also want to mention in apoptosis is that there is basophilic staining of the nucleus. So if you take the cell, if you look at the cell that is undergoing apoptosis, you notice that the cell will be staining basophilic. Once you apply your H&E stain, it's going to take the hematoxylin part and it will appear purplish, basophilic. Why? Because the nucleus is condensing. So it's like we're having a very high amount of the nuclear material. The DNA, the RNA, they are kind of constricted together. So they can readily take up those basophilic, because they are highly basophilic, they will appear blue. But the cytosol will not do that, because these changes are destroying the cytosolic component. The proteins are going. Part of what is even in the cytosol is our RNA. So we are losing RNA, we are losing other protein, and because of that, there's nothing to pick up the basophilic state. So it will appear eosinophilic. So the cytosol in apoptotic tissue, or cell rather, is eosinophilic, while the nucleus is basophilic. And the nuclear changes include pekinosis and nuclear fragmentation, karyoexis, due to the caspase. And the caspase is also going to disperse the cytoskeleton, which will lead to formation of the plebs, leading to formation of the apoptotic body that will be engulfed by the macrophages. This one will shrink.
Okay.
The cell in apoptosis shrinks. The cell in necrosis swells, because necrosis usually follows reversible to irreversible. Yeah. So the cell in necrosis will be swollen, but in apoptosis they shrink. So let's go. So just to add more information, so let's break down the cell shrinkage. There's chromatin condensation, there's membrane blebbing, formation of the apoptotic body, and they are phagocytosed by the bacteria, by the macrophage. So that means that there's no cellular content that is spilling out to drive inflammatory response. No. It is characterized by a deeply eosinophilic cytoplasm. I've said that because of the loss of all the cytoplasmic protein. And it is basophilic, with respect to the nuclear staining, because of the pekinosis, the condensation, typically. There's something I want to mention here. We can see it. Cell membrane typically remains intact without significant inflammation, unlike necrosis. That's fine. There's something we call DNA laddering. Now, the part of these nuclear changes, this caspase also activates endonuclease. So the endonuclease that it activates kind of split the DNA fragment into some multiples of 180 base pairs. So what that means is that you just notice that you're having, once you do your maybe like flow cytometry now, or like staining, you are going to notice that the DNA is going to be occurring in laddering in multiples of 180. So you can see DNA fragment that is weighing 180 base pair. You can see another one 360, 540. Multiples of 180. That is what we call DNA laddering. That is a sensitive indicator that cell is undergoing apoptosis. We can see this in question. I've seen this in a question before. I was trying to look for the question, I could not find it. So please, we must always remember, DNA laddering is due to activation of endonuclease by the caspase, which causes breaking of DNA in fragment of 180. So once you see, okay, I think I remember where I saw it. Yes. You guys remember the, what's it called? The U-word assessment that we do when you are done with UWOD. Do you guys remember those two guys?
Yeah.
I think I saw it in one of them. And one of the things is that once you do it, it doesn't stay, it will disappear. So you cannot recall, you cannot go back to it. It disappears. After like maybe like 7 days or so, or 1 week, it will go off. So you won't see it again. I saw it in one of the questions that I was doing. So DNA laddering is a clincher for apoptosis. It breaks it in multiples of 180. And don't forget the inflammatory response is not there. Yeah. So I think there's something else I wanted to say, but okay, so let's look at okay. Let's look at this question. If 46-year-old man comes to the office with slowing growing neck mass, the mass is stony hard on palpation and seems to be fixed to the adjacent tissues. After initial evaluation, combination chemotherapy is prescribed to the patient. Several weeks after, the mass significantly reduced in size and biopsy demonstrates a shrunken eosinophilic cells within the tumor. Which of the following substances released from the mitochondrion mostly trigger the observed cellular change? So what do you guys think? So someone
Cytochrome C.
Hello, you said what?
No, cytochrome C.
Cytochrome C.
Cytochrome C.
So that's very correct. So cytochrome C will be released. It will activate the caspase, which will cause the apoptosis. It will cause the death of the. So it's very easy to figure out because this guy is giving chemotherapy. What do you guys think will chemotherapy do?
The DNA damage or
It will
injury.
damage DNA. So if it damage DNA, what pathway is it going to activate?
Intrinsic
Intrinsic.
pathway.
Intrinsic. And what is
P53 activation.
exactly? So and you guys remember that when it activates the P53, what will P53 do?
That will
Activate the
activate
back
the
and
back
back.
and the
So when they
back.
activate back and back, what will happen?
Activates the
You form
what's it
pores on the
called?
mitochondrial membrane.
It will form pores
Yeah.
within the mitochondrial membrane, and what will
Yes.
escape?
Cytochrome C.
So what would be the function of cytochrome C?
To
You
activate
add
the
with
cytosolic
ATP
caspase.
and then activates the caspase.
See, that's the application to this question. So just before we leave, let me just mention that in addition to DNA, misfolded protein and hypoxia here. So in addition to DNA damage, misfolded protein and hypoxia can also trigger the intrinsic pathway. Misfolded protein, hypoxia. So if any protein is misfolded and you want to take it out, or any DNA is damaged, you want to take it out, or a cell that is hypoxic, some of them can undergo death by apoptosis. We talked about the triggers for death through the death receptor. So we're looking at negative selection in the medulla, and we're also talking about I think that's essential. Then we said damage of malignancy. Yeah, thank you, Dr. Zulia. Then we said the perforin granzyme pathway can also be used by our natural killer cells and the cytotoxic T cell. Okay, so do you have any question? I think we are going to call it a day and continue tomorrow from here. Do you have any question?
I have a question, but I don't know whether that is also apoptosis. So when we use glucocorticoids to relieve inflammatory processes or something, I think I came across it somewhere that also induces apoptosis. So like under.
Are you sure? Okay.
Yeah, I saw
it reduces apoptosis. Well, it could be. You know that the function of glucocorticoid is to inhibit NF-κB. I know that NF-κB produces all the cytokines in the body. I remember that one of the cytokines is what? TNF, Abi?
TNF alpha.
Yeah, TNF alpha.
I remember that one of the pathways for apoptosis is what? Extrinsic pathway.
TNF alpha.
Which uses TNF alpha. So that could be.
Okay. It was more like it induces apoptosis of macrophages and something like that, and then also eosinophils. So I did not really understand the correlation of how the glucocorticoid came about inducing apoptosis of macrophages and eosinophils. I was like, how did
you say induce or reduce?
Like,
Induce.
only was explaining reduce.
I
Yeah,
thought
that's
said
what
it.
induce.
So they're like,
So
IN.
so if it's induced, then that one is okay. So what's glucocorticoid? What do we call it? Is it anti-inflammatory or inflammatory?
Immunosuppressor,
It's an
anti-inflammatory.
immunosuppressant. Exactly.
Mm-hmm.
All these guys utilize interleukins in their activity. So if you cannot produce interleukins, there's nothing to stimulate the macrophages. There's nothing to stimulate the release of lymphocytes. You get. So they will not be there. I was even thinking you are trying to relate it to apoptosis. So that was what I just thought about. Do you understand, Dr. Ziliad?
No, I don't understand
I
though.
don't understand too.
Ah, okay. So let me state it again. So you guys know that if you want to recruit macrophages, what do you need? Is it not gamma interferon that you use to activate macrophages?
Mm-hmm.
Yes,
You
yes.
even need something to release your gamma interferon. You need to activate like maybe TFR1. And
Yeah.
what is even needed to activate the TFR1 is like interleukin 12, right?
12. Yeah.
All
Yes,
these
yes.
guys are cytokines. They are produced from activation of a transcription factor. What is a transcription factor? NFKB. Now, glucocorticoid will come. It will inhibit NFKB. That transcription factor is not there. There's nothing to produce those cytokines again. So that means that there's nothing to even activate our macrophages. There's no interleukins to drive inflammation again. You guys
Okay.
get. So that's why it is
Yes.
anti-inflammatory. So all your eosinophils is dependent on interleukin 5. There's no way your interleukin 5 will form if you have inhibited NFKB.
Mm.
You get. So
Yeah, but I was just wondering why, because the answer of that option was induces apoptosis in eosinophils. I'm like, I did not think about, so it
Right.
okay, so
induces
this
it.
is it.
Yeah.
This is it, Dr. Zuliad. If
Mm.
there's drop in interleukin 5 for eosinophils, what pathway of apoptosis will be activated?
If there's drop in interleukin 5.
Remember
Okay, now I get.
we talked about the regulatory pathway. We said
Mm-hmm.
interleukin
Okay, if
when
we
you
reduce regulatory
factor.
factors, Abby.
Yeah, it's because we did not see that question. If we saw the question, we'll be able to relate with it. So you know that you have taken out that regulatory factor like that. So there's nothing to stimulate those cells again. So the intrinsic pathway will now kick in.
Oh, okay.
Okay,
I
so
think
it's
I
by
can relate to that
apoptosis.
one.
I
Yeah, so
get.
that
So
one
that's
is going to be via the other. Okay, so the trigger there will be the when you remove the regulating factor. That's what
exactly,
reinduce the apoptosis.
exactly,
Okay, totally.
exactly.
Okay, I
So will
get
it be by
it.
intrinsic or extrinsic?
That
It
will
will
be
be
The
by
intrinsic.
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 intrinsic. It will
That's
be
by
by intrinsic.
intrinsic. Okay.
Yeah, thank you
All
for
right.
Okay.
sharing.
Thank you.
Thank you very much, Dr. Zulia. Okay. So I think we can call it a day, guys, so that we can continue tomorrow. So
All
Yeah.
right.
Thank you.
bye bye everybody.
Bye bye. See
Bye
you
bye.
around.
Thank you.